normal cell growth, or what in another way you can call it as a molecular basis of cancer. So the ILO for the today's lecture is, first thing you must know the basic pathology of the tumor, like what is a tumor, what is its definitions, what is benign and what is malignant tumor. Then there is demonstrate knowledge of the basic concepts of the cell proliferation and carcinogenesis. Cell proliferation in a way both normal and abnormal. But here we are not going to discuss much in detail about the cell cycle and how a cell divide. We just see how a cell undergo division and where it can get affected and can turn from a normal cell division to a abnormal cell proliferation, which in turn lead to the formation of a carcinoma. The other word, it's known as carcinogenesis or genesis of a tumor. Then you must know what is the genetic basis of the cancer or the molecular basis of a cancer. Cancer is now concept as a, the new concept for the cancer is it is a genetic disease. So there are alteration at the molecular level, at the various molecules of the genes get altered, which in turns lead to the abnormal cell proliferation and the development of a cancer. Cancer, it doesn't mean like genetic means it is always a inherited disease, okay? No. Genetic means the genes of a normal cell, but it can be of somatic cells or a germline cells, okay? So if it is in the germline cells, obviously it is inherited one, but it can happen anywhere in a person's normal life where his genes are exposed to the different mechanisms of the carcinogenesis. He can develop a cancer. So we have genetic mechanism both in sporadic as well as inherited cancers. So then you have to know a little bit about the spread and clinical effects of the tumor, how a tumor is spread and what can a tumor cause different type of clinical effects. So basic pathology of tumors. What do you mean by tumor? Actually, if you remember in your life cycle inflammation lecture, the tumor, this term was originally applied for the swelling due to any inflammation. You can recollect the five signs of inflammation like Keller, Rubor, dollar, tumor, and loss of function. Okay. So in this, the initially any swelling caused by inflammation is known as tumor. But nowadays, when you say it's a tumor, it means it is a neoplastic one. No one call a non-neoplastic swelling as a tumor. When we call it is a tumor, it is a neoplastic one. Okay. So what is a neoplasm? Okay. A neoplasm is an abnormal mass of tissue. So it is not a normal mass. Okay. It is an abnormal mass of tissue in which the growth is excessive and uncoordinated. When the normal cell divide, the division and the growth of cell is limited, not very excessive, and it divide in a coordinated matter. Okay. So where in the neoplasm, the growth is excessive and uncoordinated. Okay. And this growth is persistent. Okay. In the same excessive and uncoordinated manner, even you stop the stimulus, even after cessation of the stimuli or stoppage or the break of the stimulus. Okay. So, and this stimulus, even you break, but once it goes into the neoplastic transformation, the cells will continue to divide excessively and uncoordinated. The example is over here. We can say smoking. It is a documented fact that smoking causes cancer tumor. Now, a patient of recently diagnosed with the CA lung, okay, He has come to you and asked, Doctor, I am smoking. And as you told that my smoking might have caused this tumor. Okay. If I stop my smoking now, can my tumor also stop and it will go back to normal? No. Once the process has started, once the tumor has, once the cell has changed into a neoplastic cell, even you stop the stimulus, it will not go back to the normal. Okay. So this is a definition, a complete definition of a neoplasm. Actually, neoplasm, the true meaning of neoplasm is a new growth. But when you call it is a cancer or a tumor or a pure definition of neoplasm is excessive, uncoordinated, persistent growth which keep on growing in the same manner even you stop the stimuli which has caused this growth. Okay, so this is the definition of a neoplasm. Now, when you have come to know there is a tumor can be benign. Benign is like innocent tumor, okay? Or it can be a malignant tumor. When do you call it as a benign tumor? When the tumor grossly, when you see the tumor open eye, it doesn't look very like what do you call very destructive. There is no hemorrhage, no necrosis. Okay, tumor is well circumscribed, it is localized, it has not infiltrated the surrounding tissue. And when you see under the microscopy, it exactly look like it's benign it's normal tissue, like fibroadenoma of the breast or fibroma of the fibrous tissue or polyp or papilloma of the skin. Okay, these are the tumor, benign tumor exactly look like their original cell of origin, and they will remain localized. They don't metastasize. They don't cause much problem to the patient, and they don't cause, if they are located in the normal location, they usually don't cause the death of a patient. Okay, and they are easily curable with the surgical removal, and they don't recur. Okay, this is the definition of a benign tumor. While malignant tumor is exactly opposite to the benign. They don't resemble, most of the time they are normal cell of origin. They are very destructive in nature. They invade, infiltrate the surrounding tissue. They infiltrate the blood vessel or lymphatic, can go to the distant organ from lung to the adrenals, from breast to the bone to the lymph nodes. Okay, and they affect the patient's the function of the norm, the organ where they are located, and eventually they can lead to the death of a patient. So these are the basic pathological terminologies when we use in the tumor, like a neoplasm, the benign neoplasm, and the malignant one. Now, a brief about the etiology of the tumor. Okay, so how a tumor, like most of the time, the exact etiology of the tumor is not known. Okay, a tumor, sometimes a tumor comes in the collection of all these events. Okay, or sometimes a single event like inherited one. Okay, so like suppose a family, a person's parent, one of the parent or both parents have one kind of the tumor, the like retinoblastoma, a tumor of the retina can be seen in the newborn child. Okay, or sometime it is like suppose someone has a inherited genes. Okay, and they get sometime infections, like suppose the Epstein-Barr or EBV virus infection, they get incorporated in the gene like MYC or MYC gene and can develop Burkitt lymphoma. Okay, so this is inherited one. Then chemical. This is also very, very common, like because we are living in a chemical world. Everywhere you look around your surrounding, you have chemicals. Like our body is like we use the soap, it is chemical. We eat something like junk foods, they have preservatives, they are chemicals. Okay, someone works in the industries. Okay, some of us goes as a radiologist, they are the radiologists working in the department where they have exposed to the radiation. Okay, so it is like exposure to the environment, exposure to the surrounding. But there are few chemicals, they are very, very highly carcinogenic, like polycyclic hydrocarbons. They causes sarcomas like coal, gasolines, aromatic amines such as benzylamine, aniline. These are usually seen in the color industries. They are causes different type of carcinomas. The alkaline nitrosoamines like nitrite compounds, which you generally use as a flavoring and the preserving agents in the foods. So they can cause liver carcinomas. Okay, and there could be like some of the chemicals, these are the directly acting chemicals, some of the chemical which can act as an indirect acting. Okay, suppose a fungal infection of the aflatoxin. Okay, it usually seen in the nuts or the full sudani, usually infected with a specific type of fungus. If you ate this fungus, it will get converted in your body and from a normal aspergillus fungus to the aflatoxin B, which can cause to the different type of cancer, especially the hepatocellular carcinoma. Then there is a physical carcinogens. You go outside, there is ultraviolet rays in the sun. If you work in certain places like radiology department, which I has already explained, like ionizing radiation, someone working in the nuclear medicine or someone working in the mines to extract the nuclear material, they have exposure to the different type of carcinomas, ultraviolet rays to the skin, ionizing radiation, x-rays usually causes leukemias, lung cancers seen in the miners who are working in the mines of the radioactive material. Asbestos usually it is used in the clothes or in the roof, like where you use the different special type of roofs in the rural places. These industry workers are exposed to develop a specific tumor known as mesothelioma. Then the microbiological, you know very well, like the bacterias, one of the common bacteria which we you read in the case 2, H. pylori. There are viruses like DNA viruses, human papilloma virus, like cancer of the cervix or cancer of the skin, herpes virus. It is also especially type 2 cancer of the cervical epithelium, eh, eh, like HBV, hepatitis B virus, hepatocellular carcinoma, Epstein-Barr virus, lymphomas and leukemias, human T cell leukemia virus, leukemias, hepatitis C virus, hepatocellular carcinoma. So these are RNA viruses, these two, and these four viruses are the DNA viruses, which can cause different type of microbiological cancer, carcinogenesis and cancers. Okay, here I specifically not included bacteria, H. pylori, because they you know very well about the H. pylori. Now, fundamental principles of carcinogenesis. So, what happened to the cell whenever there is exposure to the above mentioned or the which I mentioned previously to these various type of like genetic mechanism or various chemical exposures or physiological exposure, there is the non-lethal genetic damage to the cell. What do you mean by non-lethal? Means usually whenever there is a damage to the cell, there is different mechanism which will try to repair these genetic damage, or if they are not, the body is not able to repair, they will take cell to the apoptosis, the program cell death. So the genetically damaged cell will not enter into the cell cycle. But what happened over here? The This genetic damage has caused cell to become immortal. The cell will not die. So that's why it is called non-lethal genetic damage. Okay. So that can be acquired by the various chemical or that can be inherited in the germinal indication. Okay. There are four type of very important genes. Like actually there are many genes, but the four genes are very important to be remembered for development of the carcinogenesis or genetic alteration in the genes. So what are these genes? The growth promoting oncogenes, growth inhibiting tumor suppressor genes, the genes that regulate apoptosis, and genes that involve DNA repair. As I told already to you, whenever there is a genetic damage to the cells, even the these genes that repair the DNA, they will try to repair these genetic damage, and bring a normal repaired cells into the cell cycle. Or if it is not able to repair, this gene will comes, they will make this cell to undergo apoptosis or programmed cell death, so that the abnormal cell should not come into the play, or it should not come into the cell cycle. But these genetic damage is very smart. He knows how to skip this mechanism, so an abnormal cell can go into the cell cycle and tumor can progress. Tumors are monoclonal. Always remember, whenever there is a growth, growth is polyclonal. But when there is a carcinogenesis, tumorigenesis, a formation of neoplasm, whether it is benign or malignant, it is monoclonal. What do you mean by monoclonal? There is clonal expansion of the single precursor cell. A single a damage to a single cell will enter into the cell cycle, and there is a single clonal expansion which will lead to the monoclonal growth, which we call as a neoplasm. Okay, this is a normal cell proliferation. Okay, whenever there is need of a cell to undergo division, like in a normal organogenesis in fetal or embryonic life, or whenever you have like injuries where you need the repair mechanism. So that stimuli will lead to various growth factors will come in play. These growth factor will bind to their specific receptor. Then there is activation of these growth receptor, which is for the transient and the limited time of period. Once this is required, this activation of growth factor receptor will stop. Then this activated growth factor will activate the several signal transducing protein in the inner side of a cell membrane. Usually these growth factor receptor are located on the cell membrane. So they will bring the various signals inside the cell membrane, inside the cytoplasm. This cytoplasm will send signals across the cytoplasm to the nucleus. In the nucleus, there is there is activation of the nuclear regulatory factor that initiate DNA transcription factor to enter these DNA to the cell cycle and ultimately result in the cell division. This is the normal cell proliferation. The same mechanism happen in the abnormal also, but there is everything is abnormal. The abnormal growth receptors or like hyperactivated growth factor will activate the growth factor receptor, which will not be transient. It will be like for a long period of time, and this activation will continue and will send the cell to the like in the cell cycle continuously, so that the tumor keep on growing. So, so far we are discussing about the what is the genetic. There is a genetic damage. There is a genetic damage. So, what kind of genetic damage happens in a cancer? So, remember, cancer is a genetic disease. Here, genetic disease, we don't mean it is a inherited only. The genes can be affected both in the inherited cells and in the somatic cells also. Okay. And this genetic injury, as I told, it can be inherited or can be acquired by environmental agents like various physical agents or chemical agents or microbiological agents. Okay. Then the tumor develop as a clonal expansion of the single cell, a monoclonal expansion of the genetically damaged cells. Four important class of the genes. Okay, so these four important class of gene, if you have been asked, mention four important class of gene which is involved in the molecular basis of the cancer. Okay, so you write oncogene, tumor suppressor gene, DNA repair genes, and the genes that regulates apoptosis. So there is transformation, then progression, the growth will invade the normal surrounding structure, and then it can go to the metastasis. So the cancer formation is a multi-step procedure. It is not a single step. This is a multi-step procedure. There is a growth, there is invasion, there is metastasis. Okay. So what are the hallmark of a tumor cells. Due to these genetic abnormalities, the tumor cell acquire certain hallmark feature or certain characteristic features. So what are these characteristic features? First thing, they become self-sufficient in growth signals. They don't require any other signal to send them or any other trigger to send them signal to multiply. They are self-sufficient. They send themselves signals to themselves so that they don't need any external thing. They are like self-dependent. Okay. But still our body has, if something abnormally happening to the cell, our body has growth inhibitory mechanism. So these tumor become a more smarter. They become insensitive to the growth inhibitory signals. Okay. First, they acquired the self-sufficiency. Okay. Like you, a child, take a child, like say a distorted child, not a good child. Okay. A bad child. He started earning. So he think now I self-sufficient. I will not listen to anybody to I don't require money from my father. I can sustain myself. So he is self sufficient. Now his parents are telling him not to go outside, not to drink alcohol, not to smoke, not to not to involve in illegal relationship and all those things. He will not listen to them because he is not dependent on them. So he'll become insensitive. So same happen with these cells. The cells become insensitive to the growth inhibitory signals. As well as what happen, there are like you have mutawa roaming around. He can modify you if you are not listening to your parents. But they will become both, like the DNA repair mechanism is something which is like repair beyond your parents. Okay. So they will become insensitive to the DNA repair mechanism, or they will make dysfunction in the DNA repair genes also. Even there is one more is there, a capacity. We can send them to the prison. Still they have a characteristic. They can, the cell become even more smarter. They are in become insensitive, or they can escape from the apoptosis so that they can enter into the cell cycle continuously. With these events, they also acquire one more thing, is limitless replicative capacity. What do you mean by limitless replicative capacity? We have the specific type of genes, telomerase. When they see the cell, a cell is designated only to 100 times to enter into the cell cycle. After it has achieved 100 times, telomeres comes into the time. They cut the cell, the ends of the DNA, and they will bring the stop codon. Once the stop codon comes, the cell stop division. Okay. Our cells stop multiplying. But these cells become ageless, or they cannot die. Okay. Because of this telomerases. Okay. Now the cells is growing. Okay. Still it can be stopped if it will not get nutrition. And nutrition comes from blood vessel. So they are smart enough. They get their own blood vessels. They get their own factors, different factor they secrete, and they get their own blood vessels so that they can continually get nutrition. Okay. They don't stop over here. Okay. They are very notorious. Once they have developed, they have sustained, like Israel. It has, like, once they stop small places in Palestine. Now, once they have like got their power, they started invading the areas. Okay. So they invade to the surrounding tissue. Even they don't, if they are in the liver, they don't stop in the liver. They go into the blood vessel and go to the different organs. Okay. So, and along with that, they also have the capacity to escape the immunity. Okay. So these are the hallmark of the malignant cells. Okay. So are you understanding?
Yes.
Yes,
Yes,
yes.
you're following each and every step. You have anywhere any doubt, please ask me. Okay. So this is just a depiction what happens in a tumor cell. First, they get the self-sufficiency, then they get the insensitivity to the growth signal. They have like evasion of the apoptosis. They got like defective repair mechanism, sustained angiogenesis, limitless replication, then invasion to the tissue and the blood vessel, and they keep on growing. So this is called molecular basis and hallmark feature of a tumor cell. Okay. Now we go about the genes. What are the important genes? Obviously, as I mentioned, the four important genes, each and every gene is important. Out of these, the four which I mentioned, oncogene, tumor suppressor gene, DNA repair genes, and genes that regulate apoptosis. These are four very, very important genes. Okay. In that, even these oncogenes and the tumor suppressor genes are very important. Okay. So these oncogenes are actually, they are the normal proto-oncogenes. Okay. They are nothing abnormal actually. They are proto-oncogene, which is the normal genes that stimulate the cell divisions. Okay. Tumor suppressor genes are also the normal genes which inhibit the cell division. Okay. These are the normal genes. Okay. Usually whenever there is a requirement of the cell division, proto-oncogene will stimulate the cell to undergo cell division. When the cell division work is over, there is a role of tumor suppressor gene which inhibit the cell division. The cell cycle stop. Okay. And usually they are usually seen in the repair mechanism or during the embryonic growth periods. Okay. And they are very, very strictly controlled mechanism. Okay. This mechanism of the normal growth is strictly controlled mechanism. You have different phase, G1, G0, M, S, and G2. Okay. So these are the different genes where you have the phases of the cell cycle, and these genes work over there. Okay. What happened in the tumor? Okay. In cancer cells, there is like this normal control has become defective. The same proto-oncogenes, when we call in the normal cell cycle, in carcinogenesis, they call oncogenes. Okay. In normal, there is always a balance. But whenever the tumor, there may be excessive oncogenes, so the balance will go, or there is insufficient inhibitors. So anyway, when the balance is lost, the cells will under keep on multiplying. Okay. So this is normal when there is a, it is usually seen in the normal growth repair mechanism. The stimulators, oncogenes, the inhibitors, tumor suppressor gene remain in the balance. While in the cancer cell, this balance is lost. One is more. As here you see the stimulators are more, so balance is lost. Sometimes it happens, inhibitors become abnormal. They are not able to inhibit. Okay. So it is there, inhibitors are not there. Okay. Or they are there, but they are abnormal. So the balance is lost. So this is the normal cell. Okay. Whenever the normal cell, we are continuously, our cells are exposed to the environment. Okay. Like we are continuously exposure to the chemical radiation, viruses. Okay. So these stimulus or these agents or these carcinogenic agents or these normally occurring agents in the environment is affecting our cells. They are causing our DNA repair. Okay. But Allah subhanahu wa ta'ala has given us very beautiful mechanism. So our body is continuously in the repair mechanism. Okay. So there is a normal successful DNA repair. The cell become normal and the normal cell remain normal. But whenever there is a failure to the DNA repair, okay, this failure to DNA repair comes from where? Sometime the inherited mutations in the genes, okay, or sometime persistent exposure to these, like a severe viral infection, body is not able to clear these viruses, or someone working in the chemical industries, someone working in the radiological department, these DNA fail to repair. Okay. These mutated cells of the a mutation in the genome of the somatic cells or the inherited cells, any cells can go into that. There is what happened. There is activation of the growth promoting oncogenes. There is inactivation of the tumor suppressor genes. and there is alteration in the gene that regulate apoptosis. What happened over here? Even if the cell fail to repair the DNA, these apoptotic cell can lead to the cell death. Okay, but here there has mutation has set these three mechanisms: activation of the oncogene, inactivation of the tumor suppressor gene, alteration in the genes that regulate apoptosis. So these two mechanisms will lead to unregulated cell proliferation. Okay, and this will lead to the decreased apoptosis. Okay, so these unregulated cell will go into the clonal expansion. This will get its own blood vessel. Due to the angiogenesis, it will escape the immunity. Even at this place, if the immune mechanism is very strong, it can kill the tumor cells with the help of various cytokines, various antibodies. Okay, that can stay, or like we have like tumor natural killer cells are there in our body which can kill these clonally expand cell, but it escape the immunity, it gets its own blood vessel, or it does keep on getting additional mutations. Okay, so tumor progress, it goes into the malignant neoplasm and it will invade and metastasis. So this is our molecular basis of the cancer. So what are the oncogenes? I will just explain these in brief. Okay, so these are the normal gene, but when it get mutated, we call them oncogenes. This will give to the self sufficiency to the cell to enter into a growth cycle. Okay, so what are these example? As I told, it is a normal, like we have growth factor, growth receptor, signal transducing mechanism, and the nuclear mechanism. Okay, so you have growth factors. Like this is the example of one of, there are many growth factors. Okay, so you have like one growth factor like platelet derived growth factor. It is involved, specifically involved in a one tumor of the brain known as glioblastoma. Okay, growth factor receptor. Okay, so they have like ret. This is usually involved in the multiple endocrine neoplasia type 2 and type 2b, which has familial medullary thyroid carcinoma, pancreatic carcinoma, gastrinomas, various endocrine neoplasm. Okay, then you have the receptor tyrosine kinase c-KIT. C-KIT is very specifically involved in the GIST, gastrointestinal stromal tumors. ERBB1, it is usually in the lung glioblastoma. ERB2, also known as HER2/neu or Herceptin, this you know where it is involved in the breast carcinoma. Okay. Then you have signal transduction protein. They are KRAS, HRAS, NRAS. KRAS colon and pancreas, HRAS bladder tumor, NRAS hematopoietic like leukemias and lymphomas. Then you, after the signal transducing protein goes to the nucleus. So they are nuclear regulatory protein, MYC. This MYC is involved in various type of tumor like lymphomas. Then there is NMYC for the neuroblastoma and the LMYC for the lung carcinoma, small cell cancer of the lung, specifically small cell cancer of the lung. Okay, NMYC neuroblastoma. So remember easily N N L L. Okay, and CMYC, the another type of CMYC, but Burkitt lymphoma. Then you have the cell cycle regulator, cyclins or cyclin dependent kinase. Even these cell cycle regulator get involved. We have cyclin D genes, especially in the breast, colon, esophagus, liver. We have like P16. If there is germline mutation in this cell cycle dependent kinases, P16, we get melanoma. In the somatically acquired deletion of P16, pancreatic carcinoma, glioblastoma, esophageal cancer. So these are all the oncogenes and their example. There is like only this oncogene require only one lecture, entire one lectures for the oncogenes for the explanation of different mechanism. But for you to just remember, I have selected important genes and associated important cancers. Okay, so if you have to remember, you must remember only this name and this tumor, this name, this tumor. this name, this tumor. Okay, a big thing to remember for you, but we cannot help you in this. So this is that mechanism, which this is growth factor. They are binding to their receptor. You have the signal transducer like RAS. You have nuclear regulatory proteins like cyclin D, cyclin D4, and you have like transcription factor like MYC. So this is how a normal and abnormal cell cycle happens, and when a tumor normally or abnormally can go into the cell cycle or carcinogenesis or tumorigenesis. Now we go to the tumor suppressor gene. Okay, tumor suppressor gene, as name suggests, they just switch off the cell proliferation. Okay, and which cycle they work? They work in the G1 phase. Very, very important to know. They work in the G1 phase. Okay, so, and these are the recessive genes. Okay, so we need to have loss of both copies of this gene to be required to develop a cancer. While oncogenes are the autosomal dominant genes, they are the dominant genes. Only one loss of one copy can lead to development of the tumor. While the tumor suppressor gene, they are recessive genes, so we need to have the loss of both copies. Okay. And in contrast to oncogene, these genes are loss of function genes. Okay, so you said like this is the tumor suppressor gene. Okay, so if they get activated, they will suppress more and more tumor. So here their function has to be lost. Their normal function of the suppressing a growth cycle is lost. Okay, so that's why the cell loses the sensitivity to inhibitory signal. Okay, the most important gene or like most important type of or prototype of tumor suppressor gene is retinoblastoma. But the most important tumor suppressor gene our body is the P53 gene. Okay. So what happened over here? One defective gene is inherited of retinoblastoma gene, and malignancy develop only when there is the other allele is lost due to a somatic mutation, which is known as the two-hit hypothesis. Okay, a person has born with the one function of the only one allele of the gene. Due to various environmental factor, the other allele is also lost. So this is called two-hit mechanism. So you have like there is like NF1 gene, neuro and fibromatosis gene, APC gene, adenomatous polyposis coli gene. Okay, they can be sporadic, they can be inherited. Okay, so these are the tumor suppressor gene. Prototype is retinoblastoma gene, but the most important is P53 gene. I am repeatedly telling the most important tumor suppressor gene is the P53 gene. Okay. It is located on the chromosome 17 and it is involved in various carcinoma. But I have mentioned here very few of the important where it is directly affected: lung, breast, colon, and sarcomas. Retinoblastoma gene located in the chromosome 13, retinoblastoma. NF1 gene located on the chromosome 17, neurofibroma, malignant peripheral and nerve sheath tumor. APC gene, chromosome 5, colonic carcinoma. WT1 gene, chromosome 11, Wilms tumor. So as I told, P53 is the most important gene. So we go for the P53 gene. It is known as the guardian of the genome. Okay, because this is one of the most important tumor suppressor gene in our the body. Whenever there is defective cell proliferation or when there is a DNA damage, this P53 cell will get activated. It brings two genes. One is that first it brings DNA repair mechanism genes. So it try to bring those genes to repair the DNA. If that repair is not happening, it will bring the genes to bring apoptosis so that the abnormal cell either get repair or can go into the programmed cell death. It should not enter into the cell cycle. Okay. So it is abnormality is seen in the almost 30 to 50% of the all cancer. Germline, like in the germline cells, it is known as it is cause a special type of tumor syndrome known as the Lee-Fromeney syndrome. Okay, these genes code for the nuclear phosphoprotein, which expressed in the increased amounts following cellular damage, especially in the following the radiation or chemical. As I explained, there is like anything happening to the cells, there is a DNA damage, P53 is activated, cell cycle arrest, successful repair. Okay. P53 if not activated, okay, what happened? It will no cell cycle arrest, but it will no DNA repair, but it will bring the apoptosis gene. So it will go into the program cells. Okay. But if the tumor cells P53 is not activated, it will get into the P53 dependent gene, no DNA, no cell cycle arrest, no DNA repair, no apoptosis. Mutant cells go into the clonal expansion of the monoclonal cells. Okay, so here it has activated P53 cell cycle arrest. If there is no DNA repair, it will go to the apoptotic mechanism. But if it is damaged, P53 is damaged, they will go into the loss or mutated P53 gene. This mechanism will happen. Normal P53 gene, these two mechanism will happen. Okay. Either activation, by activation, cell cycle rest, DNA, successful repair. If P53 is not activating, still the cell has capacity. If the normal P53 gene is normal, cell has capacity to bring apoptosis. But the cell who have the mutated or loss of P53, they will not bring these two mechanisms and cell will undergo malignant transformation. Now we have the fourth important gene that regulate apoptosis. These are the genes which bring apoptosis if the cell is not repaired. Okay, they will lead to the programmed cell death. But tumor cell acquire the quality to dysfunction these genes, and this is one of the very important gene is known as the BCL2 gene, which is seen in the follicular lymphoma. So BCL2, there are two questions can be made over here. Which of the following is a gene which regulate apoptosis? BCL2. Okay, or BCL2 is a type of gene, apoptosis, tumor suppressor, oncogene, DNA repair. So it's the gene that regulates apoptosis. BCL2 or expression is seen in which of the following tumor? Follicular lymphoma. Okay, then we have the genes that regulate DNA repair, because tumor cell affect, as I told you, these are the repeatedly I am telling the four important genes or four important mechanism: oncogene, tumor suppressor gene, gene that regulates the apoptosis, or gene that repairs the DNA. So this tumor cell also affect this DNA repairing gene. Okay, they can get affected and they will have different types of the cells. Okay, like mismatch repair gene, if this is affected, have the HNPCC. Nucleotide excision repair gene, xeroderma pigmentosum. Recombination repair gene, ataxia telangiectasia. BRCA1 and BRCA2, they are principally as a tumor suppressor gene, but they have a very, very important mechanism in the DNA repair. Okay, so that's why we are including them here as a DNA repair gene, and they are associated with the familial breast cancer. The another mechanism which will lead to the limitless replicative potential of a tumor cell, and this will become through the telomerases. Okay, because telomeres are the normal thing which will get shortened, leading to the massive chromosomal abnormalities and cell death. Okay, because as the cell will keep on dividing, we have the specific time. Okay, beyond that, cell will not go because of the telomeres. When they become short, and the stop codon will come, and cell will stop multiplication, and eventually it will go into the cell death process. Okay, but these genes, tumor cells are uses telomerases. Okay, these are the enzyme which help them to maintain telomere length and keep replicating. Okay, so this is the limitless replicative potential bring about by which enzyme? Telomerase. Telomerase is responsible for limitless replicative potential. Angiogenesis also, like they need nutrition, so they get their blood supply, and where from there they get blood supply? Various growth factors. Okay, like endothelial growth factor, platelet derived growth factor, or granulocyte monocyte colony stimulating factor. Granulocyte monocyte colony stimulating factor, platelet derived growth factor, or intermediate growth factors. These are the growth factors, or vascular endothelial growth factors. Okay, fibroblast growth factors. These are the growth factors which is responsible for the tumor angiogenesis. And tumor angiogenesis is not the normal angiogenesis, it is a abnormal angiogenesis, which will provide sustained nutrition for the tumor cells to grow. So as this all the mechanism you have seen through involving of the various gene, so this indicate the tumor formation is a multi-step process. You don't say like only a single gene is involved. There is a play of multiple genes. Okay. So they have like various oncogenes. They can always involvement of the tumor suppressor gene. And the example of these multi-step carcinogenesis is the adenoma carcinoma sequence in the colon cancer. Okay. It's a prototype example. What happened to the normal epithelium? It undergoes hyperplasia, which will lead to formation of a benign tumor, adenoma. These benign tumor, due to continuous these multi-step genetic abnormalities, it will go under dysplasia. This dysplasia with help goes into the carcinoma, and this carcinoma will invade and metastasize. So the tumor is a multi-step process, and this adenoma carcinoma sequence is a specific example for the multi-step carcinogenesis. This inshallah we will discuss in the next lecture. So cancer can be sporadic. Most of the time, 80 to 90% they are sporadic, and they happen due to the somatic mutation in the somatic cells. This can be spontaneous or it can be induced. Hereditary, 5 to 10% of the cancers are hereditary, and they are due to the germline mutation. So here we discuss characteristic of the hereditary cancers. Hereditary cancers often occur earlier than the sporadic cancer. Okay, suppose a person of the colorectal carcinoma sporadic cancer develop after 50 years of the age. In the hereditary cases, this colorectal carcinoma can have at the earlier age, so at the like 30 to 40 years of the age. Okay, so when you suspect a hereditary cancer, screening from the younger age is recommended. The individual who have the inherited genes may be at a higher risk of the more than one type of cancer. They will not have just one cancer. They will have multiple type of cancer like carcinoma, sarcoma, carcinoma of the colon, ovaries, endometrium, all together. Okay. So if suppose we have diagnosed the hereditary cancer and we have treated one cancer in the patient, but you need to have a very, very meticulous follow-up for this patient. Okay. And this hereditary cancer have a site specific cancer. They will not just go for anywhere. They will have the combination of organs. Okay. Like in APC, we have colon, pancreas, stomach. In HNPCC, we have like endometrium, ovaries, colon. Okay. And usually two or more relatives are affected. Okay. So what are these hereditary cancers? In for example, leishmania, it is due to the most important P53 genes, involvement of the breast cancer, soft tissue sarcoma, adrenal cortical carcinoma, brain tumor, leukemia. It is due to the germline mutation of P53 gene. Germline mutation of P10 gene will lead to the Cowden syndrome or Banyan-Zona syndrome. Okay, here we see the breast carcinoma, glioma, multiple hamartomas. These hamartomas are not tumor, but otherwise they are the presence of abnormal cells at abnormal place. Okay, like cartilages in the liver or epithelium, cartilages and fibrous tissue in the stomach. This is known as the normal tissues at the abnormal tissues. Okay, hamartoma. Hereditary breast and ovarian cancer. This is due to germline mutation in the BRCA1 and BRCA2. They have breast carcinoma, male breast carcinoma, male prostate carcinoma, ovarian tumors, okay, pancreas carcinoma, brain tumors, all this, okay. Then we have this Lynch syndrome, very important. This is due to the germline mutation of the DNA repair gene, MLH1 and MSH2, MSH3, MSH6, PMS3, colon, stomach, small bowel, bladder, okay. Multiple endocrine neoplasma type 1, meningene, thyroid, adenoma of the pituitary, pancreas. MEN2, red gene, thyroid, parathyroid, pheochromocytoma. Peutz-Jeghers syndrome, STK1, hematomas polyp in the GIT. They are non-malignant, okay, but they have tumor of the colon, small bowel, stomach, breast. So this is about our lecture about the abnormal cell proliferation. I hope we have covered all the ILOs. So now you know the basic terminologies, what is neoplasm, what is benign tumor, what is malignant tumor. You know the basic concept of the cell proliferation and what is the different etiology of the cancer, how the carcinogenesis happen. You know the molecular or genetic basis of the cancer, hallmark of the tumor cells, different genes involved in the tumor cells, how the tumor suppressor cells and genes and oncogenes balance is lost in the tumor, how like DNA repair genes and genes that regulate apoptosis, angiogenesis genes can help in the growth of a tumor. Okay, then you know about the sporadic and hereditary cancers. Okay, and spread and the clinical effects of the tumor I did not include over here. We'll see the spread of the tumor with the specific tumor. Okay, with we have like colon carcinoma. To some effect we can see in the practical also, practical slides. Okay, so this is clear for you all.
Yes, clear.
Yes,
Okay,
doctor. Clear.
so you go through the lectures, okay, and if you have something to be explained to you, please let me know. Okay, you can ask me anytime. So here we take a break for 5 minutes, and we'll see you again at 5 past 10, and then we'll start inshallah our next lecture. Okay.
Okay.